From Microbe TV, this is TWiP, This Week in Parasitism, episode 221, recorded on September 21st, 2023. I'm Vincent Racaniello, and joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello there, Vincent, and everybody else, as Daniel would say. Actually, this is a spectacular day in Fort Lee. I don't know what it's like where you guys are, but this is like a perfect day. No low humidity, temperatures in the middle 70s. And, you'd wish for 300 days a year like this. And if you wanted, you can move to San Diego because that's what it's like every day there.
Also joining us from New York, Daniel Griffin.
Hello, everyone.
And from Glasgow, Scotland, Christina Naula.
Hi, everybody. I'm calling in from a cold and soggy, dark Scotland. I've been soaked twice by the rain today. So, yeah.
Well, that's...
You Scots don't like umbrellas because of the wind, so you tend to get pretty soaked when it rains.
It's true, yes. Yeah. I've had one destroyed umbrella this week already, so
Wow.
I'm not trying that again.
We have two guests for you today who contributed our clinical case last time, and they're going to help us with the reveal. From Boston, Massachusetts, Michelle Negeli. Welcome to TWiP.
Thank you. Hello, everyone.
And from Vienna, Austria, Alexander Grieb. Welcome to TWiP.
Greetings from a warm summer's night in Vienna.
Is it really warm there?
It's about 22 Celsius. I don't know what that's in Fahrenheit, but it's very pleasant outside, I guess.
What's it like in Boston, Michelle?
It's actually quite dry today, so not bad. Not bad.
Good deal.
Before we start, remember, folks, we'd love to have your financial support to do these podcasts. You can go to microbe.tv/contribute. And we also have a new member of the Microbe TV cast, Matters Microbial by Mark Martin. It's on episode number 7. He interviews various microbiologists, 20 minutes or so, short programs. You can go to microbe.tv/mm. So what we should do, folks, let's go through the case. And then after that, perhaps we'll talk a little bit about Michelle and Alexander and talk about what you are up to, okay? Does that sound good?
Sounds good.
All right. So either of you, maybe you could split it. I don't know. Michelle, Alexander, why don't you read for us the case presentation?
You want to start, Michelle?
I can start. Let me just pull it up. Okay.
So to remind those tuning back in and those tuning in for the first time, Michelle?
Yes.
Okay. So we have a 36-year-old male who presents to the local ED with shortness of breath. Additional symptoms include stinging pain in different parts of his extremities, fatigue, abdominal cramps, and bowel irregularities. Oh my God, I can't do it today. Irregularities. On exam, he was resting comfortably and his vitals were normal, but the patient appears very distressed by symptoms. His left eye showed signs of irritation, including redness and swelling. A detailed history reveals travel to the Caribbean about 4 years ago and several short trips to Central and East Africa, all between 5 and 10 years ago. His initial labs were grossly unremarkable, and a chest X-ray showed no abnormalities. The patient himself suspected a specific parasitic etiology of his symptoms, so we performed a number of serologies and stool examinations in search of parasitic diseases. None of the tests revealed evidence of an ongoing infection. He's HIV negative and not sexually active at the moment. He has no pets and no other recent travel history. He eats a normal diet and has been out of work for 2 years. And yeah, that's the case.
All right, let's start with our case guesses. Dixon, can you start, please?
I would be happy to. Christopher writes, "Good afternoon, TWiP team. My thought for this case is schistosomiasis infection. Sometime, somewhere, our 36-year-old male patient took a dip in the shallow waters of a river or lake filled with snail hosts. The schistosomes released countless ova into his circulatory system, where some blocked in small capillaries of the lungs, eyes, skin, and bowel. As the immune system tried to wall off the eggs, neighboring organs suffered from the collateral damage. In his case, the pulmonary vasculature was injured, leading to shortness of breath either through pulmonary hypertension or heart failure. There are no other clear signs of heart failure seen on physical exam or x-ray, but I'm wondering if his abdominal pain may be caused by congestion secondary to heart failure. The conjunctivitis, stinging pain, and abdominal cramps with bowel irregularities could also be caused by damage done by the immune system's response to the eggs lodged in these tissues. The timing of these findings makes me hesitate. Unless he lives in a place where schistosomes are endemic, he might have had this infection for at least 5 years. How are multiple organ systems being simultaneously affected now and not gradually over the course of years? Is it possible we aren't finding any ova or signs of infection because the sensitivities of O&P serologies aren't 100%? Maybe the adult schistosomes have been dead for a while, so we won't find evidence because of a waning antibody response or cessation of egg production. As always, I'm left pleasantly puzzled. Cheers. Christopher.
Christina, can you take the next one, please?
Eyal writes, "Dear Vincent and the sages of the microscopic eukaryotes, greetings from Sydney and the land down under, where the temperature is a nice 20 C, as is proper for the first week of spring." I always, whenever Eyal writes, I get that song in my head from Men at Work.
Really?
Yes, it's from a land down under,
Yeah.
but I can't remember the rest of the lyrics. Anyway, I just had to throw that in because it always pops in my mind. For the case study
Men
this
at
week,
work.
Men at Work, yep.
Yeah.
For the case study this week, I will have to go only by the written description of the case, as was sent in by Michelle and Alexander. Unfortunately, the sound for the podcast was cut off at exactly 1 hour and 15 minutes, so I can't use Dr. Griffin's tone and innuendos to help my guess. Unfortunately, for me, not the patient, the initial lab results were grossly unremarkable and a chest X-ray showed no abnormalities. Also, as noted, the patient's own stool examination had no interesting results. Based on this lack of lab results, I've decided to try and match common Caribbean parasite disease symptoms with the patient's reported symptoms of shortness of breath, stinging pain in different parts of his extremities, fatigue, abdominal cramps, and bowel irregularities. That was a difficult word for both of us, Michelle. I've ruled out schistosomiasis as fever and rash are typical symptoms but are not presented by the patient. Lymphatic filariasis was ruled out as there is no swelling. Whipworm was ruled out as I would expect this to show in the stool examination and would not cause pain in the extremities. Paragonomyosis, caused by the lung fluke, was ruled out as there was no fever or hives. Chagas disease was ruled out as the timeline is too short for major neurological damage and there are no signs of heart problems. The only disease I could find that matched the symptoms to some extent was ascariasis. The pain in the extremities might have been caused by some duct blockage. I have no idea. So my guess for this week is ascaris and could be treated with albendazole, mebendazole. On a different note, in the last week, there has been an interesting case of larva migrans caused by Ophidascaris robertsi, ascarid found in an Australian woman's brain. First document the case of the carpet python parasite in humans. I was wondering if you could review the case study in the coming episodes, and he sends a link for the episode or for an article, 2 articles, and says many thanks for all that you do. Eyal, Did you not discuss that case, Daniel, in another podcast?
Yeah, it's so funny. I was thinking about it. I discussed it in the clinical update. I discussed it in the ID Puscast, but I didn't discuss it on TWiP.
So we're now in the process of revising the 7th edition to go to the 8th edition, and I've been assigned the nematodes, and I just reviewed that case yesterday. So, yeah, it's an interesting case. She was 64 years old. She lived north of Sydney. She lived along a lake that she used to take a walk around, and apparently there are a lot of carpet pythons in the area. And somehow she became contaminated with an egg from an ascaris inside one of them. And she actually had a biopsy done to make the diagnosis, and she survived very nicely, and so there's no side effects.
Yeah, more than they removed. You can actually see photos of the worm.
That's right. It's pretty impressive.
So Dixon, how did she get it? The egg? She ingested an egg? Is that the idea?
Yeah, the 3rd stage larva is inside the egg, and that's what she ate.
And then how did it get
by accident? By accident, and then it would migrate in the body because once the worm gets inside the tissues, it's got chemoreceptors to tell it which host it's in, and in this case, it said, "Hey, this doesn't taste at all like a carpet python. I'm sorry, I have to get the hell out of here." So, and then they try to get the hell out of there, and they go all over the body. And this is not just for this ascaris; it's for Toxicara, Canis, and Toxicara cati as well. The dog and cat ascarids do the same thing, and they all end up in the brain or the eye because those are immunologically less likely to react against the parasite. Let's put it that way.
So when you first ingest it, where does the development occur? In the stomach?
Well, in a python, it would actually come out of the egg into the lumen of the small intestine and grow up to an adult.
Okay. So in the human, it comes out and then it says, "This is not a python."
This doesn't taste at all like a python.
And so it burrows into the circulatory system?
That's right. It goes through the gut tract and gets into the blood supply, and then it could end up anywhere, basically.
Wow. Pretty creepy.
Creepy. And then as a note, I'm sure you're going to take that out of the text, but it says this is ultimate proof that nature abhors a vacuum. I mean, we probably have been infected with virtually every infectious disease agent on this planet. And not all of them cause disease in us, of course, but some do.
Daniel, can you take the next one?
Certainly. James writes, this is an interesting one. A 36-year-old with a bunch of nonspecific symptoms. Suspects parasite. It is TWiP after all. And somewhat remote travel to Caribbean and Africa. We don't know the patient's home country for sure, Germany or Austria from the Vienna Passion Club. What's true and what's a red herring in this case? Is it a tropical parasite or is it a locally acquired one? Stinging pain in extremities makes me wonder about B12 deficiency, which can be related to Diphyllobothrium, also would give fatigue, confusion, depression, also can have vision problems. We could get that tapeworm almost anywhere. I might expect the stool O&P to be positive, but don't really know the sensitivity specificity. Labs might be expected to show a macrocytic anemia with B12 deficiency. Let's see, how many dozen parasites can cause bowel irregularities and abdominal cramps? Amoeba, Giardia, worms, long list. Eye irritation, of course, brings to mind Onchocerca, but sometimes also Loa. Guess I'll go with the tapeworm and B12 deficiency. Got to get back to my intro to micro lectures. James M. Small.
Michelle, can you take the next one?
I can. So, Rafid writes, hello, unfortunately I had trouble downloading the last 20 minutes of the last episode, so I hope that I'm presenting the right case. As a clinician, if I would have seen this patient in the emergency room with bowel irregularities, shooting pains in his limbs, and who has a list of things that they want tested, my first thought would be a functional syndrome, such as irritable bowel syndrome with fibromyalgia, which would be the most likely diagnosis. Luckily, your podcast forces me to think a little deeper. The first parasite syndrome that I think of that links dyspnea, a change in bowel habits, and peripheral neurological symptoms is chronic Chagas. I'm not sure what to make of the ocular symptoms, but I did find some case reports of uveitis in chronic Chagas. I would start my investigations with an echocardiogram to see if there is any evidence of cardiac involvement, and I would do some stool tests for blood. Fecal calprotectin to rule out inflammatory bowel disease. If the cardiac echo shows LV dysfunction, I would test for Chagas. There seem to be many ELISA tests out there, so I'm curious to know which ones the TWiP experts would choose. I would like to make another shout to Michael Libman in the Tropical Disease Department at McGill. I would highly recommend for a teaching award. Thank you for your great work, Rafid.
Alex, can you take the next one?
Yes, I'll try. Reem writes, Dear TWiPers, hi, my name is Reem and I'm currently a sophomore in high school. I first heard of Dixon Despommier when I encountered his book, People, Parasites and Plowshares, which I can also recommend, when organizing a huge stack of books at the Columbia University Press. As payment, I was allowed to take that book along with many others centering on biology home. After reading the first chapter, I became obsessed with parasites, only to later discover that my mom has been working with Dixon to publish his most recent book. Shortly after, I discovered the podcast and have been listening avidly since then. Sadly, I'm only about 20 episodes in since the episodes are so long and I have to focus on school. Anyway, I just wanted to say thanks for making the podcasts. Also, does anyone have advice for how to get volunteering opportunities at labs? I've tried to reach out to multiple labs hoping for opportunities, but it's hard to be taken seriously as a 15-year-old. Sincerely, Reem.
It's not even a guess. I'm sorry. It's a fun letter.
It's
A
hard,
nice email.
nonetheless.
Nice email, though.
It's a very nice email, and I just didn't pay attention, obviously, when I was pasting it in. But yes, and Reem, the problem is most institutions, you have to be a certain age to work in a lab. And I think 15, at least in the U.S., is too young. I don't know about in Austria or in the U.K. Probably it's the same, right, Christina? How old do you have to be to work in a lab?
I would say 16 at least.
Yeah. All right. Paul writes, my top guess, toxocariasis. Rationale: 1, unilateral eye disease. 2, a parasite with worldwide distribution seems more likely given short and remote travel history. All right. Back to the top, Dixon.
Martha writes, Dear TWiPers, again, I am probably responding too late to episode 220. Well, no, actually. If we had been able to ask questions, I would have asked why the patient thought that his problem was parasitic. Has he been treated for a parasitic infection in the past, or perhaps failed to take a prescribed treatment? Nevertheless, I will jump in with a guess. He had traveled to areas where he might have acquired an interesting parasite 4 to 10 years ago. The lifespan of Wuchereria bancrofti is said to be 5 to 8 years. While alive, the worms interfere with the host's immune response. The dead and dying worms no longer exert this effect on the host, resulting in an inflammatory immune response. I can imagine that the patient's symptoms are due to this. I'm keeping this short. Best wishes to all, Martha. P.S. I've already won a book. Thanks. However, I still enjoy doing the puzzle. I like it.
Christina.
Okay, the University of Central Lancashire Parasitology Club writes, oh, that's the first in a while. I was wondering where they were. Dear TWiP professors, greetings from the University of Central Lancashire Parasitology Club. We have been extremely busy with exams and a successful graduation for some of our group. There's my answer. And we are now starting our 2023-24 academic year with excitement and vigour for the challenge of this new case. Our candidate list for the case included schistosomiasis, manzanellosis, and babesiosis. The general fatigue suggests a systemic involvement, but our interest was piqued by the involvement of the eye and the travel history to Africa and the Caribbean, suggesting filariasis. Manzanella ozardi is a filarial parasite transmitted by biting bitches in South America, the Caribbean, and Central and West Africa. In Infections may be asymptomatic, but case reports indicate that fatigue, respiratory symptoms, joint pains, and vague abdominal symptoms. Eye involvement can lead to swelling, sometimes referred to as a Ugandan or Kampala eye, and this is perhaps why the patient suggested his own parasitic diagnosis. It would be quite funny if he wrote in to answer the case on TWiP. Diagnosis relies on detecting the parasites in peripheral blood or skin snips, which might explain the negative finding on serology and fecal examination. Treatment with ivermectin appears to be effective and is fascinated that effective treatment of manzanellosis might include doxycycline to kill the endosymbiont Wolbachia. We pin our hopes on Manzanella, and as always, we are grateful for the challenging case and the opportunity for learning. We eagerly await the arrival of Parasitic Diseases Volume 7, and we will send a photo of the group when the book arrives. All the best. Neil Vickers and Neil Poyas, on behalf of the University of Central Lancashire Parasitology Club.
All right. That does it. A lot of these people have won books. We only have 3 people who haven't won a book yet. It's very interesting. All right, Daniel, what should we do next?
I guess now we let the group take some guesses.
All right. Well,
so who wants to go first? Dixon, you want to go first?
Well, if you insist.
Like that little parasitic joke there. I'm going to insist.
Yeah. Perhaps I should actually. Roll up in a ball and stay in the corner for a while. Until I heard the word Manzanella ozardi, I was kind of in a fog as to what this might be, because there are very few parasites which actually exhibit this kind of broad spectrum of vague illness, and yet are quite diagnosable once you know something about their travel history. And you look for it. Of course, to look for it means you've got to do a specialized blood test. And I'm not sure that was done. So unless it was done, and if it was, then perhaps I will withdraw my guess. But my guess, of all the guesses that were made from all the write-ins, Manzanella ozardi, for me, appears to be the most reasonable.
Okay,
but
Christina, do you want to go next?
Oh, I'm going to make such a fool of myself. I've just, literally, I just recorded my Manzanellosis lecture, and I just opened it up, and I'm not sure. So I've been trying to exclude things. So you've done a serology test, which showed nothing, as far as I can tell from the case description. So we do have filarial serology tests. We have Chagas serology tests. So I wasn't sure. Yeah, I was finding it really difficult. What else went through my mind? Let me bring up my note, because I was puzzling over this all afternoon. So also filarial infection, actually the transmission efficiency is really, really low. So obviously I don't know how long that particular patient stayed in any of those potential regions. So maybe not a filarial infection. And then I thought, maybe the stinging pains in the extremities, maybe that's a peripheral neuropathy maybe. So let's think about what could be hosting in the brain. That could maybe also explain bowel issues if the autonomic nerves are involved. So I'm really just, shooting an arrow in the dark here. So maybe a larval infection. I don't know, Toxocara, someone mentioned that. Sparganosis briefly came to mind. But I have to say, I just really don't know. I think I would need to find out a little more. So I'm going to admit I don't know, and I'm not sure.
All right, Vincent.
Well, I like this Kampala eye, so that's Manzanella. Because otherwise it's very hard to pinpoint, but this little bit of local lore, which gave me that clue, I think that's Manzanella, like Dixon said. Yeah. What about you, Daniel?
Yeah, I don't know. So this will be me guessing as well. So I would have liked more information, but that's okay. And I also I settled on Manzanella as my most likely, but of course I had a couple thoughts. And also I was going to actually say Manzanella perstans, by the way, just to pick a different subspecies, and I'll tell you why in a second. But we get that there were a bunch of tests done, but I don't know how often in the bunch of tests someone would necessarily do a filarial serology, which sometimes can be positive, even though you're really gearing those tests for other filarial species, but sometimes it can be positive in this case. The reason, and maybe someone can correct me, with the sort of different subspecies of Manzanella, there's Ozardi, but there's also the perstans, which actually really is the Ugandan or Kampala eyeworm subspecies. And this can actually get you these painless conjunctival nodules, eyelid swelling. So, if you do a more careful exam, you might see that. How do you diagnose any of these? You can do the microscopy, the visualization of the microfilarial in a blood sphere, but, concentration techniques, so you often miss it. You can, as I mentioned, get that cross reactivity. A little bit of the difference here is I would like to know a little bit more about where this individual did travel, because if I was considering the Manzanella species, then it kind of matters a little bit. The Ozardi, you're going to treat with ivermectin. If you're worried about perstans, then there's actually some little bit of a difference between whether they acquired it in Mozambique or the DRC, where you might actually treat with doxycycline, because interesting enough, Manzanella is in that world of some of them, but not all of the regional ones actually have the endosymbiont Wolbachia. Okay, so now I always like to make sure when I'm wrong, I've talked a whole lot and dug myself a super deep hole. So that was what I was left with.
All right, so Michelle and Alexander, fill us in. What's happening here?
So, I think I need to apologize a bit for the case because it's a bit of a curveball. But this is a disease that's rarely, if ever, mentioned in parasitic disease lectures or books. But I personally encounter it quite frequently in clinical practice. So this gentleman was sent to the ED where I have been working for the past 6 months at the moment from the urgent care center, where he presented with these symptoms, and he told my colleague there that he was all these symptoms were caused by schistosomiasis. And when he presented, of course, we did all these examinations and sent him on to the infectious disease clinic, where, of course, all the examinations, stool examinations, etc., were done. Filariasis, so the tests for the filarial parasites in the blood were also done, and a whole lot of other testing. And it all came back negative. And when we elicited a more detailed history, it turned out that he had also sought treatment at 2 large academical centers in Germany. And there, just as with us, everything came back negative. And because of that, he came to us because he felt that the doctors there were incompetent and did not take him seriously. So when we presented him with these findings and suggested the diagnosis that we have found in this case, he was very unhappy. He declined further treatment and left us. And the disease we're talking about today is called delusional parasitosis. I think Dr. Griffin is intimately familiar.
No, these people never come and see me. Just joking.
And I think,
this is quite a frequent thing that happens when,
And I think Michelle can tell us a bit more about what we know about this disease in general, if that's all right, if that's what you...
Yeah.
Okay, yeah. So now that we've established that this patient had delusional parasitosis, this is something that, as Alex mentioned, that we see quite frequently. That is not mentioned too much in lectures. So we thought we think this is kind of an interesting case. So what can I tell you about delusional parasitosis? There's not a lot of epidemiological data on this topic, but it's a somatic type of delusional disorder that's usually monosymptomatic. There's primary and secondary delusional parasitosis, or DP for short. The primary one arises spontaneously, and the secondary form usually occurs due to another major medical or neurological or psychiatric disorder. And patients are mostly well-educated Caucasian women, mostly middle-aged or older. And this often happens after a stressful life event. So this could actually be after the patient already had a parasite prior and is very stressed to maybe still have that, or that it's come back. Or it could be something totally different, like losing a family member. Exactly. So the source of this imaginary infestation, because basically what delusional parasitosis is, a patient really is 100% sure that they have this parasite, but there is just no evidence of this parasite. It's kind of, you'd have to exclude everything before giving this diagnosis. You have to make sure that the patient doesn't really have a parasitic disease, and you have to make sure that you're on the right track here. So, usually every doctor's visit starts kind of the same. There's definitely symptoms there that are very serious and real to patients, which can include like very itchy skin and all kinds of symptoms. And then later when the patient's giving the history, you often notice that they have been checked for multiple things. There have been multiple stool exams. There have been blood tests. There's everything in the books. And unfortunately, sometimes you just have to come to this diagnosis. And then I just want to talk about two signs or two things that I find quite interesting, or we find quite interesting. Two things you often can find in this group of patients is something called the matchbox sign, which is when patients bring in specimen in little matchboxes or bags or bottles, and they seal it airtight. And they are sure that there's like evidence of this parasite in there. And oftentimes this is just hair, fibers, coagulated blood, or might just be legs from some completely harmless insect. And that occurs in about 26% of the reported cases. And then there's something else, which is called folie à deux, which just means shared mental illness. So a lot of times patients who have this delusional parasitosis will mention that other family members, for instance, or people living in the home are also experiencing these symptoms. And oftentimes when the patient A, the inducer, gets treated, magically all the symptoms from the other people also resolve. And the hardest part of this is, as Alex tried to mention, is treatment, because usually if you mention the word delusional or psychiatric to a patient, they try to get out really fast, or they think you're incompetent. But I think it's important to treat patients seriously, to be empathetic and take it very seriously. And in non-severe cases, a good relationship as a physician or as a parasitologist and entomologist with the patients can help the healing, or can actually resolve the symptoms. In more severe cases, psychopharmacological medication may be important. Yeah, to say a little bit about this diagnosis.
It's interesting that
I'm so glad I didn't settle on a diagnosis now.
They look good. And we all did. So
yeah, I suppose you did say the patient himself suspected a specific parasitic etiology of his symptoms, and that may have that's probably the best hint.
Yeah.
Now looking back, with 2020 hindsight.
Well, also, if, as you said, no evidence of an ongoing infection. Is there any parasitic infection that would cause symptoms yet have no evidence of an ongoing infection?
I guess what you can sometimes find in patients is like secondary skin lesions from scratching, but you won't actually find the primary skin lesion that a parasite that infects the skin would maybe give you.
A lot of the patients will bring in bugs of various kinds that they can find in their local environment.
Yeah.
And I was a technician first before I became a bona fide parasitologist, and the lab was clinically very competent, and we got, I would guess, one or two of these cases every month. And they were all different people, of course. The same ones didn't come back, because we said, well, we've looked, and here, we'll look at the microscope together and I'll show you. There's the real parasite, and here's what you brought in that you thought was the parasite. And so you can see the difference right away. And they look at you and they say, "Oh, you mean I'm cured?" And I said, "Yes, you're cured." And they would walk out, and I'm sure they would go on to the next diagnostic lab and look for a better diagnosis. Because of this show, this podcast, Daniel and I have been inundated with people sending us, now that it's easy to send images, that you can actually take with your cell phone and then make enlargements and then say, "Doctor, what have I got?" And I'll say, "Well, you've got delusional parasitosis." And it's quite obvious. They don't want to hear that.
The Matchbox has been replaced with the iPhone photo.
That's right. That's right.
Can you make that diagnosis, though, without a psychiatrist or a psychologist, an evaluation?
Well, that's a great question. Technically, you would probably need it. You can only suspect it. But in the published literature and also in my personal experience, way less than even 10% of patients accept referral to psychology, or even sometimes you try to frame it in a different way. You tell them that they may need some help coping with their symptoms and with the struggle for a diagnosis. And even then, they usually refuse. And even if that's something that some people do, that's not very ethical, but some people sneak in antipsychotic medication and tell them it's to help them relieve their symptoms, which is true because some antipsychotics have sedating effects. But even that, most of the time, is not really accepted because people really want antiparasitic treatment for the parasitic disease. And a detail that I've omitted in this case is that this patient had also already received several courses of praziquantel and also ivermectin, albendazole. It only helped relieve his symptoms for 1 or 2 weeks, and then the symptoms came back. And he insisted that he needed more treatment, which is also a good indication that there is no parasitic disease going on here, because he is probably sampled most of what the Pyxis has to offer.
Just this week, I got several of these delusional. Some of them are quite long, as Daniel and Dixon have said. And one person said, she's been listening for years and gave evidence for that, cited several episodes that have been listened to, and really begged for help, saying, I really, please answer this email. Please don't ignore it.
Yeah.
And then another one here, it says, I have beetles coming from my gums, teeth, nose, and stool. I've had live flies come out when I was on the toilet. Get those all the time. And
oh, wow.
It's very sad, isn't it? Because you can't really just send them packing. You do. I mean, it would be really challenging.
If you're in medical school and you teach a long time, and I did for 38 years, I can't tell you how many medical students would come up to me after a particular lecture and say, I really think I have that.
Oh, I think that every day, Dixon. I have one I'm writing a lecture about.
Week, they get something else, and that goes away, and something else comes in and takes its place. It's psychologically. But Alexander, it's quite remarkable that you are picking this syndrome, and here you are sitting in the epicenter of the invention of psychiatry, because that's where Sigmund Freud first started his practice. So I wonder if in his early days he encountered some of this.
I'm sure.
Yeah, absolutely. Mm-hmm. And it's still going on today. And there's so much more to fear because we know so much more. So they can rattle off these names as though they were actually teaching this subject, and give you all the signs and symptoms and show you where the parasite came from and everything else. It's rather sad to see someone bothered to that extent in their life that they can't concentrate on the real issues of just being an adult. It's hard enough to do that, I can guarantee, as the oldest person here in this group at least. I'm still struggling with that concept.
There must be psychiatrists who treat these patients.
There are, but they don't want to go to them.
Well, I understand, but many people who have psychiatric issues don't want to go to
No, they
anyone,
don't.
and
No,
you have to
that's
find a
exactly
way to
right.
get them. That's part of the process, right?
That's right. That's right. That's true.
There's actually some published data from the psychiatric department of the University of Vienna. Vienna is a city of almost 2 million people, and I think they have less than one case per year, because people, even if they're referred, they really do not want to go to a psychiatrist. And I think, at least in my experience, at some point the symptoms abate. That's at least what the literature says. And at that point, people get better and they don't feel the need. At that point, when they understand that it was a delusion, they don't really require psychiatric evaluation or help anymore. So it's probably not something that psychiatry encounters too often.
Yeah.
Yeah, I think you mentioned the 10% will actually even agree to see a psychiatrist if this is what it comes down to, and maybe that's an optimistic number, but that tends to be they come to you because they know they're waiting for you to confirm, and if you don't confirm, they're going to look for the next person, and some of these people engage in self-mutilation. They're digging, trying to dig up the parasites to put them on these. And so sometimes it's a challenge. Like, how do you manage, verifying to their level that you're not finding something, getting them to stop the self-harm that's often involved. This is a really difficult area, and unfortunately there's still a lot of stigma. I mean, these are people that most providers just want to get them out of the office, so it's really hard to engage, connect with these people and help them, and particularly because in many ways they're often coming to the wrong provider, someone who's trained in parasitology, infectious disease, when actually the expertise that would help them most is the expertise they're not comfortable engaging with.
Right. And then there are the clinical cases that involve subterfuge, and by that I mean there is an underlying cause for their illness, and they have a variety of diagnosable infections which are caused by an underlying syndrome, like AIDS, for instance. Mm-hmm. And in the beginning, when AIDS first came in, it was thought to be caused by amoebae. Entamoeba histolytica was the major diagnosis for that, and they were blaming that on, of course, all the wrong reasons. And of course, we all know what happened afterwards, and it immunosuppresses and makes you susceptible to almost all infections. Then, which is why Vincent has that good question at the end of every case: what their sexual preference are and whether they've been diagnosed with HIV.
Yeah.
Which.
Yeah, but I think this is pretty compelling when you get the rest of the history. They've been doctor shopping, as we call it. They've gotten lots of treatments in the past without resolution. Nothing has been diagnostic. Yeah, there's that point when you have to just say, listen, we've looked for everything. We're not finding anything.
Right.
Are there equivalent syndromes for bacterial infections, fungal infections, and viral?
Yeah. Systemic candidemia. I mean, there's all kinds of delusional infections out there, and two-born every minute to take their money, to chelate them, to cup them, to do all kinds of stuff. I had a woman in the hospital just getting discharged today, some sort of odd therapy that involved cupping of her back and micro acupuncture that, resulted in a pretty significant real infection, which she didn't actually have prior to the. So yeah, it's really tough out there.
Daniel, I understand first-year medical students think they have everything, right?
They do.
Oh, my daughter just started nursing school, and it already started. It's like, that's fine, Daisy. You'll have everything. Right. Next week, tell me what you're studying, and I'll call it a script for that.
Well, I think this was a great case to present, Michelle and Alexander, because it's just not just another case. It's something different, and it's a serious problem that needs emphasis. So thank you for doing that. That's really great.
And they stumped the stars.
Yeah, they did.
Thank you for having us.
But I think that was the goal, right? Is for us to be like, this is not easy. Nothing's obvious.
Yeah. So that's right.
And that's when you should start thinking like, okay, even though it's on TWiP, maybe it's not.
So let's hear a little bit about you and the first Vienna Parasitology Passion Club. So Michelle, tell us your background. What's your history?
My history is I'm also from Vienna, like Alex, and I completed medical school in Vienna last year. And then I came to start my postdoc here in Boston at Beth Israel Deaconess Medical School, Medical Center and Harvard Medical School. And we focus in the lab that I'm at, we focus on antimicrobial resistance in gram-negative bacteria. So a little different, but parasites have been a huge interest of mine since basically always. I have a lot of family in South Africa, so infectious diseases, parasitic diseases are an interesting thing there. And then I found Alex at a very cool infectious diseases department in Vienna, and we found this shared interest.
So what do you plan to do when you finish the postdoc?
I would love to do residency in infectious diseases, but we'll see how that goes. Yeah.
So Alex, what's your history?
Well, I also studied medicine in Vienna, and I completed my studies 1 year before Michelle. And I've taken up residency here. Well, I'm an infectious disease resident in Vienna, which is a subspecialty of internal medicine in Austria, and it takes 6 years. And I've just completed about 1.5 years. I've spent the last 6 months at the emergency department of the Medical University of Vienna, because as part of our curriculum, we have to do several other internal medicine subspecialties, and this is where I also encountered the case. And yes, we met at the Klinik Favoriten, where I'm a resident, and I would love for her to also be a resident at some point in the future, because we share this interest, and also with a couple of other of our ID residents and physicians. And we do see some parasitic infections, so that's just paradise for anyone who's really interested in this.
So what's the first Vienna Parasitology Passion Club? Is it just the two of you, or are there other people involved?
Well, it's basically a glorious name that we give to a shared Google Doc where we draft our letters that we And when we met at the clinic, she had already planned going to the US to do her postdoc, and this was a way for us to keep in touch. And because, as you know, sometimes this can be difficult when we both have so much to do. But this sort of gives a structure to every month listen to the episode and communicate and talk about the case. And we've been doing it ever since she went to Harvard, and I'm looking forward to every episode of TWiP, and this is a great way of staying connected.
Cool.
Yeah, it's definitely a highlight of our month every time.
Well, great. Glad to hear it. And if you ever encounter another, maybe a real case, we'd love to hear. We'd love to hear from you.
Something a little easier. Come on.
Yeah, something easier. That's right.
Absolutely.
Well, I just wanted to say thank you, because I've been thinking about this all afternoon, and it's kind of kept my mind busy, you know. I thought that was really fun.
You're welcome.
All right, Dixon, tell us about the paper you've selected.
I will. So the paper I've selected, let me just pull it up on my screen here. Here we go. The title of this paper it's from PLOS Neglected Tropical Diseases. It's called Host Cell RAP1B Mediates Cyclic AMP-Dependent Invasion by Trypanosoma cruzi. The authors are Gabrielle Ferry, Daniel Musikant, and Martin Edrieria. And they're from Buenos Aires, and from the laboratory that investigates trypanosomiasis. What could be more epicentric for studying Chagas disease than where it was discovered, mainly in Brazil? So Brazil spends a lot of time with this because of the following statistics that I've copied out of the CDC bulletin. It turns out that today there are over 6 million people infected with Trypanosoma cruzi. There are 300,000 people living within the United States that are infected with this parasite. And annually, there are about 12,000 people who die from this. So it's not a trivial disease at all. And it's a very, very difficult disease to deal with because the treatment is almost worse than the infection itself. It's filled with toxicities and partial cures. and long-term disability. So there's a deep interest in knowing all the details about the life of this parasite so they can start to identify particular chemotherapies that might interrupt its life cycle. So I selected this one, not knowing, of course, that it was filled with alphabet soup. So when I say alphabet soup, I mean there are so many abbreviated cellular components that are referred to that I had to look them all up. Of course I did. And I understood a lot of them from the past, particularly RAP, which is a related RAS gene, which controls cell division, and which under certain conditions, if it's mutated, can result in cancer. So this is a surface protein that's found on all cells. And it's linked to guanosine triphosphate. So it's a phosphorylated donor in the cascade that results eventually in signal transduction and delivers a message to the nucleus to start division. That's under normal conditions. Under abnormal conditions, of course, the RAS gene is unregulated and it causes uncontrolled growth of cells. So this parasite has somehow derived the evolutionary trick of taking advantage of this surface protein, together with a certain other downstream effectors. And I will now read you the author summary, which is very simple. It says, as an obligate intracellular parasite, T. cruzi needs to gain access to the host cell cytosol. Now, for those that are not familiar with the life cycle of trypanosomes, Trypanosoma cruzi actually, when the reduvid bug, and that's Vincent's favorite word, by the way, reduvid, when the reduvid bug deposits its digested blood meal near the site of the bite of the wound, it's not transmitted by the trypanosomes going through the mouth parts of the insect. It's transmitted by you rubbing the trypomastigotes, which are found in the stool of the bug, into the bite wound. And that's how they gain entrance into the host in a natural infection. But once inside, then what happens? The trypomastigote has to somehow become intracellular because that's its life. It's in direct contrast to African trypanosomes, which are extracellular all the way through their life cycle. So you've got these two trypanosomes, which about 175 million years ago were probably the same parasite. And as those two continents split apart during the Jurassic period, the trypanosome that ended up in South America became quite different than the one that stayed in Africa. The one in Africa is transmitted by tsetse flies, but the one in South America and Central America, and even in some parts of the southern part of the United States, are transmitted by reduvids. They are incredibly promiscuous with regards to their host range. So in South America, virtually every mammal is susceptible to Trypanosoma cruzi, including, of course, humans. So this group wants to know everything that you can know about how this organism gains entry into the cell. Because if you can stop that, you can stop the infection. And that's why this is an important paper. So I'll read again. An obligate intracellular parasite, T. cruzi needs to gain access to the host cytosol. Now, when I say cytosol, I mean this is not included into a vacuole and then taken into the cell. This organism penetrates directly into the cytoplasm of the infected cell, and it becomes essentially an organelle. And organelles are not attacked by host immune systems. This parasite is totally protected from all of the normal immunological and innate responses that would protect a cell from infection. Trypanosoma cruzi crawls into the cytoplasm, and it's hiding in plain sight, basically.
Dixon, so when it penetrates, does it become encased in a membrane? Or
no, that's the point.
How does it get through the membrane then?
That's what this paper is all about. Thank you for asking. Because if we don't know that, then it's a mystery as to how that happens, right?
Yeah.
Most organisms, even viruses that you know of, are all included into some kind of a vacuole. And then things happen after that. In the case of Trypanosoma cruzi, there is no vacuole. There's no parasitophorous vacuole. The parasite gains interest into naked cytoplasm. And once it's there, it is as at home as the mitochondria, the lysosomes, the peroxisomes, the nucleus. All of those, and in fact, all of those are membrane bound. Trypanosoma cruzi remains in its life as an unmembrane bound parasite. And at that point, when they go inside the cell, the triple mastigote rapidly changes. It morphs into the amastigote form. The mastigote refers to the flagella at the end of this parasite. Once the triple mastigote goes into the cell, it loses its flagellum and develops into what looks like leishmania. Looks exactly like leishmania. In fact, they're called LD bodies. For Leishmania donovani, they look exactly like leishmania. And then they replicate. Okay, and in replication, they then overwhelm the cell, and the adjacent cells now become susceptible. And now the way the parasite moves from cell to cell is quite different than the way the triple mastigote originally enters the cell to begin with. So that's why in this paper they discuss various ways in which the parasite invades cells. Because once it's an intracellular parasite, its mode of transmission is totally different. Well, you also have to ask, well, then how does the reduvid bug become infected? And it's interesting because some of these parasites can revert back to the triple mastigote stage in blood as they're released from these infected cells. And they are now circulating in the blood of the host. And the reduvid bug, when it takes a blood meal, can suck them up. And that starts their infection. So that's completing the infection from host to invertebrate, intermediate host. So here's what else they write. The process of invasion is mediated by the activation of different signaling pathways in the host. Activation of cyclic AMP signaling has been implicated in invasion, with high intracellular levels of cyclic AMP associated with an increase in invasion. In this regard, we have recently shown that capital EPAC, which stands for exchange protein activated by cyclic AMP, and .pKa, protein kinase A, mediates the effects of the cyclic nucleotide on parasite internalization. That means that there's no phosphorylation involved in the invasion process. And in fact, in this paper, we'll see if you phosphorylate with pKa, you actually inhibit invasion. In this work, in order to shed some light into the detailed mechanisms of invasion mediated by cyclic AMP/EPAC, we have explored the activation of downstream effectors of this pathway during host cell infection. Our results established that RAP1B, a known effector of EPAC, and MEEK/ERK, and I'll tell you what those are later, are downstream effectors of the pathway. In addition, we found a pKa-dependent inhibition of infection, presumably by phosphorylation of RAP1B and potentially of EPAC as well. So it's a pretty involved description of some rather simple, straightforward experiments involving virocells. They're all of these results are obtained from parasites that were grown in vitro. So they're not replicating the natural pathway by which this parasite infects cells. They're taking the epimastigotes, which would redifferentiate from an infected cell that had the amastigote stage. When it breaks open in culture, those organisms revert back to the triple mastigote stage. And it's that stage that they can collect and allow to divide so they can have a lot of them to work with. And so that stage actually is not exactly quite like the same stage as present in the feces of the reduvid bug, but it's close enough. And it's the best they can do with regards to how they deal with this parasite in vitro. So they have invasion assays, and they have pull down experiments where they're looking for activated molecules. Um, they're, all their assays are described to begin with, and then they go into their experiments. So the results, let's start with cyclic AMP/EPAC activation as a ubiquitous mechanism of invasion in T. cruzi. So what they do is they infect some cells, and originally they've been described from NRK cells. I don't know what NRK stands for, actually. Does anybody?
I did look it up, but let me see. And I actually note it down as well. Let me just find. You carry on, and I'll come up once I find it.
But it's a cell line that's available, obviously.
I think they're rat kidney cells.
Yeah.
The NRK is a rat kidney cell.
Norwegian rat kidney cells, maybe?
Yeah.
In order to assess...
Yes, Norwegian, that's right, yeah.
It's Rattus norvegicus, that's the actual name of the animal. In order to assess the ubiquity of the cyclic AMP/EPAC pathway, they chose other cell lines. So they chose two that, Vincent, you're quite familiar with, HeLa and HL1 cells. Consistent with this result, the result which they found using a lookalike molecule called 8-bromocyclic AMP, they could then trace that molecule to find out what happens to it once they give it to an infected cell. This is amplified, and they get a lot of this produced, indicating that the infection is either associated with an increase in cyclic AMP, or as a result of the infection, cyclic AMP increases. So we're not sure whether cyclic AMP is necessary for invasion or whether invasion induces cyclic AMP. So they have to further distinguish that. So then they move on to look at RAP1B as a mediator of the cyclic AMP/EPAC dependent invasion. And they do so by inhibiting the RAP1 molecule and then showing what happens to invasion. And it's decreased when you inhibit the normal RAP1. So that the decrease, however, wasn't to zero. They got some decrease when they inhibited that molecule, but they didn't eliminate the parasite's ability to get inside the cell just because RAP1B was inhibited. So there are other factors, and that's what leads them to these other downstream molecules as potential co-effectors of infection, I should say. The next thing they did was a series of related, very sophisticated molecular manipulations of using this tracer molecule for cyclic AMP to find out what happens to it during infection and whether or not enhancement of that molecule by amplifying it before they infect results in a greater infection, and it did. So they concluded that cyclic AMP is an increase in cyclic AMP is necessary for an increased infectivity of the parasite, proving that the molecule is essential for its invasion portion of life cycle. And then the role of protein kinase A-dependent RAP1B phosphorylation. This was a little trickier for me to explain because in one instance, well, actually both of these instances, using HeLa or HL1 cells, the results look as though they had just copied the results and labeled each one with a different heading. I mean, they looked almost identical in terms of the result. The result, the invasion assay, was that if you transfect HeLa and HL1 cells with RAP1, the cells were infected to a greater extent than if you had not done the transfection rather. And so that means that more of something in some cases turns out to be a good thing. That's not always the case, but in this case it is. Now, the one I like the best, though, is the next picture that they show of indirect immunofluorescence of a normal cell on the left and an attempted infected cell on the right. And what they've shown here using antibody against RAP1B, is that there's an accumulation of RAP1B at the site of penetration along the membrane. So RAP1B is universally is equally distributed throughout the membrane of a normal cell. And the moment the parasite touches the membrane, there's a shift in distribution, and it all accumulates at the point of attack. And that's fantastic. You can't get a better look at what happens early on than the picture that you're looking at, and it's remarkable, quite a remarkable finding, I think. It's so clear cut.
So I'm trying to reconcile this, Dixon. Right?
Yeah, you go ahead.
You can tell me my issue.
Go in there.
So there's a really, these, Gabrielle and Martin have just done great work, and they have a really great paper in 2021. All roads lead to cytosol, and they actually have really nice figures. And the whole concept that they've put together is that, the initial penetration is actually into a lysosome, and then there's a leaving of the lysosome, right? That's going to occur. What they refer to as an exocytosis. And then, if you read kind of this whole idea, is that what we're seeing here is all this signaling and calcium-dependent lysosomal exocytosis is going to happen sort of in a late-stage lysosome. So I'm sort of curious, why is it all, clustered up here as lysosome is entering the cell? Because I would think what I would want to see is the signal right before it exits into the cytosol from the lysosome.
Right.
So I wasn't sure I understood that.
It might happen very quickly. I mean, this is a rapid process, okay? This might happen over less than a second, perhaps.
I guess that's the challenge. How long does it take to go from an early to a late lysosome? I sort of thought there was some sort of time period, not just seconds, but, my cellular biology is now, But anyway.
No.
Yeah, because it's an interesting process. The whole idea is there's two ways that it can get, there's two pathways to get into the lysosome. Now it's in the lysosome, but it's got to get into the cytosol for its, maturation, the next part of its life cycle. So it was just the timing issue I'm trying to figure out.
The lysosome
so those of you closer to this than us, Michelle and Alexander, what is the time differential between an early and a late lysosome?
I wouldn't know. I'm sorry.
I like to ask it on rounds. Those of you closer to this than
Right.
What is the time difference?
Listen, I spent 3 years in the laboratory of a guy who got the Nobel Prize for working on lysosomes. I should know the answer to that, but I don't think they called them early and late lysosomes at that point.
Yeah, what's the time difference?
Well, they're not interested in that, actually.
It's probably minutes, Daniel, right?
Yeah, I was thinking it was minutes, so it's odd
hours
to me to see it at the point when it's entering. What is it doing, clustering there? Unless maybe, I don't know.
No, this picture is not of the lysosomes. The red that you see there, that is RAP1B.
Yeah, and why am I seeing it at what looks like the surface?
Yeah, plasma.
Why am I?
The parasite.
Right? See the redness there?
Mm-hmm.
No, that's right. No. So the interesting biological interpretation is that direct contact of the parasite with the membrane causes the mobility of this protein to accumulate at the point of attack. Mm-hmm. So you're going to suggest something, I hope, and that is this parasite must be secreting something right there. There must be something coming out of the parasite which gives a chemical signal to the cell's membrane to migrate all of its RAP1B to the point of entry.
It could be
because at that point,
it could be a receptor interaction, Dixon. Right?
Yeah, that's right. I'm going to tell you right now that the next move is for the cell to accumulate all of the cyclic AMP right underneath that red. Mm. And this parasite has an energy source waiting for it as it gets into the cytosol. I don't think it's in the lysosome for very long. And it's got a protein on the outside, by the way, which is called it's a 63K protein, which is resistant to lysosomal digestion. So this parasite has an ability to actually avoid lysosomal digestion by not only altering the pH of the local environment, but also with a protective coating on the outside of itself, which prevents enzymatic attack. So this has evolved an enormously sophisticated strategy sounds like a virus. It's got lots of things going for it in which, without the host's cooperation, you won't get an infection.
Yeah.
And that was the attraction for me. I mean, the next couple of experiments were more or less icing on the cake to show that it doesn't involve pKa. And so pKa, it says pKa dependent phosphorylation effect on parasite release. It's a negative effect. It actually reduces infectivity. It doesn't increase phosphorylation. Phosphorylation does not increase it. So it's not part of the normal phosphorylation cascade, which would start at the outside from a signal transduction event and then eventually reach the nucleus of the host. This parasite has no interest in amplifying the number of cells that it can infect because it has its pick of all of them.
So it sounds like it does get encased in a membrane when it comes in. It looks like endocytosis.
Very, very initially, and then it's got to get out of that lysosome to get into the cytosol.
Have you ever watched these things under the microscope?
No, I have never.
They're incredibly active. I mean, they're absolutely, I've seen them in blood, and they're absolutely remarkable in terms of their activity. There's no way to mistake them from anything else. So, I can imagine these things go, and it's in, and you've missed the event. But everything happens really fast. Yeah. I mean, really fast. So, imagining...
I was going to say, but I do agree. This is really important work because currently our treatment for Chagas is just... I sometimes worry
barbaric.
Why we bother. Yeah,
It's barbaric.
Yeah, it's like 2 to 3 months of being on a medicine that no one wants to be on, and
that's correct.
The data is not even particularly compelling that we're making a difference in a lot of these cases.
And there are 6 million people out there waiting for a treatment.
Yeah.
So that's the bottom line is that
and hundreds of thousands of folks right here. This isn't just a neglected disease in some other part of the world. It's right here.
And you can transmit this quite easily by transfusion. And I don't know what they're doing, Daniel. Do you know what the blood banks are doing about this?
I don't know actually what we do here in the U.S. for T. cruzi blood bank screening. I'll take a look.
I can tell you that there was a couple that taught at NYU, the nuisance swags, and you probably
it looks like they actually might do. It looks like there's a U.S. blood supply screening. Since 2006. Actually, I know this because this is why a lot of people get referred my way, right? They go, they donate blood, they get a positive, they get a letter, we found something and you should go speak to an ID doc about what it is. And then I see them, right? And then, remember I was complaining about this? Yeah, I was like, it's all coming back to me now. But my fog, because I'm like all focused on the molecular. But this back to my daily world. I was complaining. My office was turning into a Chagas clinic because you can imagine all these individuals, 300,000 individuals, all they have to do is go and give blood. And some of the communities here on Long Island, it's like 15% of the individuals are serology positive. So they go get blood, they get a letter, they see me, we do a couple confirmatory tests, and then I scratch my head about really, do I really want to offer you benzoninol or nifurtimox, or what am I going to do?
No, you don't want to do that. No, you
because I'm not sure, again, how much it makes a difference in these late stage.
That's right. The Nussenswags, Victor Nussenswag, when he was still in Brazil, that's where they came from originally, invented a method for actually curing blood supplies of Trypanosoma cruzi by using a 1 in 4,000 dilution of gentian violet. And the blood was blue, not red. And if the blood was blue, people would take it. If the blood was red, they wouldn't.
Interesting.
And it killed off the trypanomastogotes without doing any harm to the patients whatsoever.
Yeah, here we screen, and then, that just doesn't go into the blood supply. But
I mean, you could, right. At any rate, this is a fascinating paper. I'm sorry I didn't do the other paper, though, because I would have had fun showing you why I don't like it.
Well, maybe for the future, because it's got all those things that you like to talk about. So
it does.
Yeah. Actually, it's got flaws, but it's really interesting for elucidating a lot about trichinella and stichosomes and stuff.
If you didn't know anything about the structure of the worm, it's a great paper. But there was one. Let me just say one thing.
Even the fluorescence, right? Everything was green. You're like, okay, show me how you got
That's exactly right. There was no localization whatsoever. It was everywhere. But but the kicker was they had one figure with 6 different panels in it, and it was all photographed at the same magnification. All of it. That's what they said. All of these were so that they're all to scale. Then they had one panel that had a 3-day-old adult female worm. And they had another panel that had a 6-day-old female worm. And the 6-day-old female worm was twice as long as the 3-day-old female. And that's not right. That's wrong. The female worm doesn't grow after they become females. And so I was highly suspicious of that figure at that point.
Only someone with your experience, Dixon, would know that.
No, you can go to the Racaniello page and see for yourself.
The Racaniello page is written by you, Dixon.
Well, but only if they get a textbook, and we didn't give out any textbooks this time. So
we could if you want.
We still need to.
Well, we
haven't done that yet.
We have 3 people, so let's do that.
The odds are pretty good now.
By the way, Dixon, thank you very much for that paper.
You're welcome.
All right, so
thank you.
Let's do a number.
You brought me right back to 1996, Dixon, when I started painstakingly sequencing by hand an adenylate cyclase in Trypanosoma brucei. So a large part of my PhD I spent working with Trypanosome adenylate cyclases. So it brought me right back. Loading the gel, pipetting your sequencing reactions into 4 different color tubes. So thank you. Set me down some nice memories too.
Michelle and Alex, have you won a book? I don't remember.
We have a book.
Well, we've won a book, yes.
All right.
Very good. All right. So,
but you would need 2 books at some point.
I think
we can talk about that.
Need one from the library.
We'd love the one at some point.
Yeah, we can do that. No problem.
All right. So do you really need a drum roll for this?
Give me one number between 1 and 3, Dixon.
Here we go.
Number 1. That's James.
Way to go, James.
Who knows? Maybe James already has a book. But James,
you know what?
From Rocky Vista University, you're the winner. Send us your address. You're in Colorado. That's easy. TWiP at microbe.tv. And if you already have a book, let us know. All right, time for another case. Daniel, you have anything left?
So this is actually a case from a colleague of mine, and this colleague will be joining us at the American Society of Tropical Medicine meeting in Chicago. I think it's in Chicago. If it's not, I'll be in the wrong town. But this is a great case. So this is a case of a man in his 50s. This person actually has a parasite, by the way. Just spoiler up front. No remarkable prior medical history. He is receiving care at a hospital in Northern California. After experiencing a generalized seizure. They do an MRI, magnetic resonance imaging, of his brain, demonstrating a solitary left temporal lobe T2 hyperintensity with gadolinium rim enhancement and surrounding edema. So he's got basically a lesion in his temporal lobe. They give him some steroids, some medicine to keep him from seizing, and they transfer him to the local academic medical center. Examination by the neurology consultants noted disorientation, inattention, moderate aphasia. They don't really clarify whether it's receptive or expressive, but he's having difficulty communicating, mild right hemiparesis. They do a lumbar puncture and get some cerebral spinal fluid, which reveals increased nucleated cells up to 80. Less than 5 would be normal, or ideally 0. These are 60% lymphocytes, 17% neutrophils, 23% monocytes. That does add to 100. Protein concentration of 38, which is not elevated. Glucose concentration of 100. Be nice to know the serum reference. They go ahead and do a brain biopsy of the left temporal lobe lesion. They send this off for cultures, which do not grow bacteria. They do not grow fungi. They do not grow mycobacterium. They perform metagenomic next generation sequencing on a CSF sample. They send the brain biopsy samples for universal broad range PCR for bacteria, fungi, mycobacterium, tuberculosis, non-tuberculosis mycobacterium. The pathology is initially read as showing well-formed granulomata with acute inflammation. But re-review of the neuropathology, and actually got a chance to see the neuropathology, there are these round, potentially infectious forms, based upon, there's no scale, right? So I'm using nuclei to give a scale saying about 50 microns in size with some surrounding clearing. And in the center of these roundish forms is a dark area, about half the size of the nucleus of the surrounding cells.
All right, so we can't ask any questions, right, Daniel?
That's all I got. You got what you got. I feel like I've given you enough. Yeah. I feel like you guys.
I've got all I need.
Yeah, I think people already should know what we're thinking about here.
Yeah, I still like that.
But we could ask, you could
ask stuff, and I could just say things like, I don't know.
No, I'm not going to say that. That's not worth it.
When you say round, you mean perfectly round?
No, not perfectly round. I didn't want to use the other term. I was just going to say roundish.
Oh, that's enough said.
All right.
Got it.
That'll do it for TWiP 221. You can find the show notes at microbe.tv/TWiP. If you want to take a guess at this case, which apparently everybody knows, without further clue, you can send your guesses to TWiP at microbe.tv. And if you like our work and would like to support science communication, please go to microbe.tv/contribute. Our guest today from Beth Israel Deaconess Medical Center in Boston, Massachusetts, and Harvard Medical School, Michelle Negeli. Thank you for joining us, Michelle.
Thank you so much.
And from
it's really great being here.
The Favoriten Clinic in Vienna, Austria, Alexander Grieb. Thank you for joining us as well.
Absolutely, my pleasure.
And I'm looking forward to getting your emails in the next couple of weeks to guess on this case, right?
Turnabout is fair play.
Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Thank you.
I was just up at Vagelos, Daniel. I was teaching second-year students, and Ben, he's an infectious disease fellow. Ben.
Yeah, Nico.
Yes. He's a course director, and he said he knows you.
Okay, yeah. Say hello to Ben next time you see him.
I'll see him next year.
Now, I will probably see him first.
And it's funny. So this is second year. It's microbiology and infectious diseases. I gave 2 basic virology lectures. 2 of the students in the class took my virology course as an undergraduate 2 years ago.
Oh, wow.
And they still let him into the medical school.
Let him in. My students have a good record of getting into medical school. Very good. Dixon Despommier is at trichinella.org and thelivingriver.org. Thank you, Dixon.
And Parasites Without Borders.
Now, you can't be 3 places. You can't be 3 places.
Sure I can.
No, I'm the boss. You
Watch
cannot.
me.
Thank you, Dixon. Thank you, Dixon.
You're welcome.
Christina Naula is at the University of Glasgow. Thank you, Christina.
Thank you. I had a really good time. Interesting case, fantastic paper, good company. So thank you very much.
I am Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP, and Ronald Jenkins for the music. So I think one of our guests should do this.
They should both do it together.
Oh, that's challenging.
It'll be
Yeah,
entertaining.
We can't do it.
Okay, so I read the beginning, as you know, and you two guys are going to both say another TWiP is parasitic, okay, when I'm done. You ready?
Okay.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon.
When another TWiP
Another TWiP is
is
parasitic.
parasitic.